This is a video guide on how to use CheckEM. This how-to video will cover 4 main parts. Firstly, what data do you need, the types of data you can upload, and how to format it. Secondly, how to upload the data to CheckEM. Then I'll go over how to read the quality control assessments in CheckEM. Lastly, how do you download a list of all the potential errors flagged by CheckEM, and how to download a clean or summarized version of the data. So how do you format your data? You need 2 things to upload data to CheckEM. The first is the sample metadata, which is a spreadsheet that includes all the information on where your samples were collected, what date and time they were collected, what was the water depth, etc. The second thing you need is your annotation data. How many fish did you see in those samples, how big were they, and which sample were they in? The prefix for all of the files you upload to CheckEM must be named with a campaign ID, and this campaign must be consistently named. So the sample metadata is a spreadsheet that has 1 row per sample. It has a set of required columns. It is important that all of these are spelt correctly, and some columns have a particular format that's needed, such as date time. This is the first set of columns that you need for the metadata. So opcode and period are used to define a sample. You only have to include opcode or period if you have used it in your annotation data. So if you've annotated a BRUV dataset in EventMeasure and you've put your sample name in as the opcode, then you will, and you haven't used any periods, you will only need the opcode column in your metadata. If you've done a stereo dove or a stereo ruv and you've used opcode and period to define your sample, such as transx, then you need to include opcode and period in your sample metadata. If you've used a drop camera as your sampling method and you have multiple samples within an EMOB and you've used period to define your sample, then you only need to include period in your metadata, sample metadata. Latitude and longitude are both in decimal degrees. Date time is year, month, day. T is just defining the start of the time, which is hours, minutes, seconds. And this TZD is the time zone designator. So if you're giving your date time in UTC, then you can put Z. Or if you're, say, giving your data in Perth, in Western Australia, you would put plus 0800 because we're 8 hours after UTC. Those 5 columns are all required. Site is not required. Location is not required. Status, which is if it's in a marine protected area or if it's no take, it can either be fished or no take or the IUCN level, IUCN category. Water depth in meters. Successful count is was the sample successfully annotated for count, or will that data be used in your analysis? It's compulsory, but it can be blank. Successful length, was that sample successfully annotated for length and will that data be used in the analysis? So if you have a BRUV, say, that flips upside down and you don't want to use that in your analysis, you would put it as successful count equals no and successful length no. Observer count and observer length, what was the full name of the analyst who annotated the count and annotated the length? Visibility and inclusion probability are not required, but they're both floating point numbers, so just a number that's not an integer, so it has a decimal place. This is just a video showing an example sample metadata file. So I'm just going to open it up in Excel. In this example, we've used opcode to define the sample in EventMeasure. We've got the latitude, longitude, date, time. All of this data was collected in Perth, Western Australia, so the time zone designator is +8. We've left site and location blank. All of the samples were in a no-take national park zone, so the status is no-take. The depth is all in meters. Successful count and successful length was all yes, and we filled out the full name of the analyst for observer count and observer length. There are 2 types of annotation data that you can upload. The first is an EventMeasure database output. So this comes directly from the emobs or the EventMeasure saved files. And the second is a generic type of upload. And you would use this if you do not use EventMeasure or you have data that was annotated before EventMeasure existed. But you would also use this if you did use EventMeasure, you exported the data and then you made changes to that data in a text editor like Excel, because this changed data is now the true copy of the data. To get an EventMeasure database output, you save all of your emobs in a folder. You create a directory or a folder where you want the database output to be saved. You open up EventMeasure, go to Program, click Generate Database Output. In the first entry box, you add the directory where all of your emobs are saved. In the second box, this is where you want to save the database outputs. You put the directory that you just created where you want the database output to be saved. Then add the campaign ID as the base name, and this needs to be spelt the same as the prefix on the metadata file. This is our directory in the example that has all of our emobs. So you can see down the bottom there's 64 items, so one for every EventMeasure observation file and one for the autosave. We've got that metadata sheet that we went through before in the same folder. It's all formatted correctly. So we open up EventMeasure, go to Program, Generate Database Output. In the first box, you put the folder where your EventMeasure files are saved, or the emobs. So in this example, ours are just saved in a folder called EventMeasure. You add the output file directory, so where you want the EventMeasure database outputs to be saved. And you add a base name, which is the same as the campaign ID. Then click Process. It will take a second to run. Cool. And so it's used 32 EventMeasure files and there's no errors showing. If we go back to the folder that we created, now all of the exports are saved in that file. If you do not use EventMeasure or you have data that was collected before EventMeasure, you'll need to use the generic type of upload. You'll also need to do a generic upload if you used EventMeasure to annotate your videos, but then edited the data that was exported in Excel or another text editor, as the changed data is now the true copy. The count file has opcode or period to define the sample, family, genus, species, the count, so that's the number of that family, genus, species that you saw in that sample, the stage, the development stage, so if it's adult, female, male, juvenile, or it can be blank, stage is not required, and code, so that's either a cab code or an API ID from Worms, and that column is not required either. The length file is essentially the same thing, sample, family, genus, species, count, length in millimeters, stage, and code. So this is an example, just the first 5 lines of a count and a length file. So in both these examples, the opcode has been used to define a sample. There's the family, genus, species of the fish species seen, count, stage, and code. In the length data frame, length in millimeters of that individual is also included. Now that you have your data formatted, you can start to upload it to CheckEM. This is what CheckEM looks like. This is the landing page. We recommend using Google Chrome for best performance. So the first thing you need to do when uploading data to CheckEM is set the format. First, you choose the type of upload. So did you use EventMeasure or is it generic data? Then the type of sampling method you used. So was the sampling platform in a single point, like a stereo bruv or a drop camera? So something that was stationary, or was it something that was moving and doing transects? The reason that you have to specify this is single point methods will create a max-n, whereas transect-based methods won't create a max-n in CheckEM. Next, how did you record the sample name in EventMeasure? So did you use, for a single point, did you use opcode or did you use period? This should match in your metadata as well. You only have the column in the metadata if you used it to define a sample. Or for transact, did you use opcode and period? So entered half of the sample name into the opcode in the information fields and the other half into the period, such as transact 1, transact 2, transact 3? Or did you just use the period name to record the sample? Uploading a single point campaign that used opcodes, you'll get asked, did you use periods to standardize the sampling duration? So for example, if you're uploading a stereo bruvs campaign where you wanted the system to be on the floor for 60 minutes and you set a period start time and a period end time, you would be able to work out using CheckEM which ones were longer than 60 minutes, which ones were shorter, and which ones were the correct time. Once we've set the format of the data, we choose the directory where our sample metadata and EventMeasure exports or generic data are saved. So we navigate to the folder, click upload. It will ask you if you trust this site. Click upload. And after a few seconds, it will tell you that it's uploading the data. And then the data that's shown in this preview window will change to the data that you have uploaded. If you receive an error here, it's probably because you haven't formatted your metadata following the requirements. So double check the format of your metadata. Next, we need to set which life history information and marine spatial planning and marine regions we'll use. CheckEM uses a spreadsheet of life history information to check your annotations against. This life history information includes the correct spelling of scientific names, which regions different species occur in, their maximum length or body size, and the length-weight equation parameters used to calculate biomass for that species. There are two life history lists that you can choose from in CheckEM, an Australian scale or a global scale. The information for the two lists come from different sources. In the Australian list, the scientific names are based off CAB, and in the global list, the scientific names are based off fish base and worms. The information on what species occur in which regions in Australia is from CAB and worms. For data outside of Australia, it comes from fish base and worms. All of the information on maximum size and length weight equations comes from fish base for both the Australian and the global list. The Australian list uses Australia's marine regions, and the global list uses the FAO major fishing areas. The two lists also are at different scales. So the Australian list uses Australia's marine bioregions. The global list uses the FAO major fishing areas. Next, we need to set which life history information and marine spatial planning and marine regions we'll use. Status is a required column in the sample metadata, but CheckEM does have the ability to generate one for you using the shapefiles. So the shapefiles for Australian datasets are from CAPAD, and the shapefiles for global datasets uses the World Database on Protected Areas. There are some issues with both of those, so we suggest using the column that you have uploaded, but if you are struggling to find out if your samples are within a no-take area, then you can generate them from the shapefiles, but just be cautious of them. Next, you set which life history list to use. So either the Australian list with the Australian marine regions or the global list with the FAO major fishing areas. Once you've chosen your life history list and Australian marine regions, it will bind the marine region to the metadata. It can either do this by getting one marine region per campaign or one marine region per sample. Doing one marine region per campaign is a lot quicker. It just uses the average latitude and longitude of a campaign, which is fine if all of your samples are within one marine region. But if your samples cross a large scale, such as a country or the west coast of WA, it's crossing multiple marine regions. The species observed in those regions will be different, so you want one marine region per sample so you can identify better which species have been observed that have not been observed in that marine region before. Now I'm going to go over how to read the quality control assessments produced by CheckEM. Each quality control assessment in CheckEM is displayed as a box, and the color of the box tells the user what they need to do about the potential error. So a blue box is just general information, which could be the number of samples or the number of fish observed. A green box means that the data you have uploaded has passed the quality control assessment. So you might have 0 samples that are missing metadata or 0 samples that are missing fish. An orange box means that it is potentially a problem, but it could be okay. You should review the check, such as a sample that is missing fish. It could be that there was no fish in that sample, or it could be a mismatch in the metadata. A red box means that the data did not pass the quality control assessment and needs to be reviewed, such as a fish outside of the predefined sampling range. We've set the format of the data that we're uploading, we've set which life history information we've wanted, and we've selected the directory where our sample metadata and annotation data is. So now we're going to check our metadata and periods. So it will take a second to load Australia's marine spatial zoning because it's large. This is what it will look like once it's loaded. This first quality control box is the sample metadata score, which we'll explain a little bit more later. The second box is the number of unique samples in the metadata. This third box is the number of samples that do not have any points data. This could be because no fish were observed in that sample, or it could be that the annotation data for that sample is missing. Same as samples without links. It could be that there was no fish observed in that sample, or that annotation data is missing. So in both of these examples, there are no samples without links and no samples without points. Every sample in this campaign observed a fish. All the samples in our points file and our links and 3D points file have metadata. There is none that are missing metadata. All of our periods have an end. All samples have a period. All of the points are within a period, and all of the links are inside a period. This is kind of what you want this screen to look like, with a lot of greens. The only yellow that we have is a period that's not 60 minutes long. So to look at that, we can click on the box and see that it was opcode 31, period 1 was only 53 minutes. So all of these boxes are clickable to look at which opcode or period has the error or potential error. On the check metadata tab, a map's also displayed, just showing where the samples are located. Each of the samples you can click and see the sample metadata for that site, for that sample. It also displays the Australian marine parks, which you can turn on and off. Cool. So next we're going to create and check maxn from the points file. So in this campaign, we saw just over 4,000 individuals. 11 points didn't have a number associated with them. So this is usually an analyst seeing something that's not a fish that they haven't been able to ID. They will just add a point, add a comment, and remove the number. This is an issue because Global Archive requires every point to have a number. Next, we can look at the species names that have been updated. So in this example, this ray brevicordata has changed genus to Bathytosha. This wrasse has changed from Lineolata to Lineolatus. Obliquus has changed family, and so has Sex fasciatus. We can also look at species that have not been observed in that area before. So this is using the life history list. This uses the marine regions that fish base or worms or cab says that species is present in. It's automatically set to show the unique species per campaign. You can untick this and it will show you all the samples or all the opcodes or periods. You can also filter out SPT1s and SPT2s because they're not included in the life history sheet. And then you get a smaller list of all the species that you haven't seen in that region before. You can also check the species that are not included in the life history list at all. So these are usually either spelling mistakes or the family, genus, or species has been updated but hasn't been included in the synonyms list yet. Next, we can plot the most abundant species in the campaign. So you can use this number input to change how many species are plotted. So we could look at 5 or 25. We can also plot the abundance of each species spatially. This drop-down is ordered by the most abundant species. You can hover over the circles to see how many were in that sample. CheckEM also creates some bar plots by status or by zone. In this example, we only have data from within the no-take zone. And it also does it by location and site, but this data is missing location and site. Next, we'll check the length and the 3D points. So the first two boxes just show you the number of lengths and 3D points that were measured. None of the lengths are missing a number in this campaign, but one of the 3D points is missing a number, which was a sync point, so we can go back and fix that up. 6 names have been updated, 14 species have not been observed in the area before, and 10 species are not in the life history list. So that's the same as what was in the maxen. Next, we want to check the lengths against the maximum on FishBase. So we want to know everything that's bigger than 100% of the max. So in this example, we've measured this fish as 544 millimeters, but the maximum size on FishBase is 400 millimeters. So we want to go back to that length measurement and make sure that it's actually 544. It could be that the FishBase maximum length is too small, and then we can edit that, or it could be that there's a mistake in the measurement. So we don't want to just know if a fish is bigger than its maximum size. We also want to know if it's nearing that maximum size. So we can also look at fish that are bigger than 85%. And you can use this table just to go further through that and look at all the fish that are bigger than 85% of the maximum. We also want to know if fish are too small. So we use 15% of the FishBase maximum. So in this example, Western king wrasse, the maximum size is 400, so 15% of that is 60 mil. But this fish is only 29 millimeters, so very small, and so we probably want to go back to the EMOBs and double check that measurement. Okay, next we can check the RMS. The recommended length rules for RMS is 20. But say if we drop that down to 10, we could see that there's 58 measurements over that new RMS limit. So we can look at those here, and we can put that back. Now they're all fine. We could also do the same for the precision to length ratio, or for the range. So say we couldn't see 10 meters, we could drop it down to 8, and now we have 2 fish that are further away than 8 meters. So this one is 8.7 meters and this one's 8.8. Another way of visualizing the lengths that are over the correct size is through this histogram. 15% of the FishBase maximum is here, 85 is here, and this red line is the FishBase maximum. So we can see that for Western king wrasse, it's tapering off, and there's a few that are over this 85 and one that's over this FishBase maximum. So we want to be checking anything that's over this line and anything that's under this line. We could also do this histogram by status. So you'd have a plot for no-take and a plot for fish, or for zone and a box plot for zone and status. Next, we want to compare the maxen to the total number in the length and 3D points. So in this example, there's 8 maxens where the maxen does not match the total number measured, so length measurements and 3D point measurements. So we want to go back to their EMOBs and make sure that they are equal. Because of that, this campaign has got a slightly lower score in the number in count versus number in length. And around 75% of the measurements in this campaign are length. In this plot, there's a dot for every maxen. So this is elongatus in one sample and elongatus in another sample, and you want all your dots to be on this red line where the max n is equal to the stereo max n, or the number of length measurements plus the number of 3D points. If your dots are below the line, it means that your max n is higher than the length. And if your dots are above the line, it means that your length max n, or your length measurements and 3D points measurements, is over the max n, is greater than the max n. We can also look at the ratio of 3D points to lengths per sample. The length data is usually the most important data that comes out of a stereo video dataset. We want to see a higher proportion of green in these plots. If you have any pink bars, it means that your max n hasn't been fully measured. You can also facet this by observer lengths to see if there's any patterns in the different length analysts that you have annotating your data. We can also view this as a proportion, which makes it a bit easier to compare samples. So in this one, in this example, 23 has only 25% of the measurements have length, whereas in these samples, most of the measurements are lengths. We can also look at these plots for individual species, which is important if you only measure target species. So we could look at pink snapper. All the dots are on the line, which is good. And most of the measurements are length measurements, except for in this one sample that had one fish which was 3D pointed. Now we have checked our data, we can download a list of all the potential errors, and we can also download the clean and summarized data. Add a project name or a campaign ID if you're only uploading one campaign. We set the period time if we've used it, the range limit, which has updated from when we entered it before, the RMS, and the precision to length ratio limit. We click download all errors in emobs, and it will save a CSV. If we open that up in Excel, it gives us the campaign, the opcode, period, family, genus, species, where the potential error is. It's also ordered by opcode and period, so you can check one emob at a time, and you can filter the errors to you can filter out the errors if you're not interested in, say, ones that are too small. Once you've gone back through all your emobs and you've made all your changes, we recommend that you go back through this whole process and check everything again to make sure that you haven't missed anything. And then if you want to download a tidy summarized version of the data, you can use this step 3 and step 4. So choose if you want to keep the synonyms that have been updated. You can remove the species that have not been observed in the area before. You can add in zeros where a species isn't present in that campaign. And you can choose to remove the CheckEM generated columns such as zone and marine region. You can also filter out the length measurements that are smaller or larger than the fish base maximum, and you can remove the 3D points and length measurements that are greater than the range limit, RMS limit, and precision to length ratio limit. Once you've selected all your filters, you click this download all files, and it will download a zipped folder. If you've chosen to add in the zeros, you will get a row for every single species in every single sample, with a 0 if that species wasn't present, and the max n if it was. And you only get these 3 files, count, length, and mass. If you If you don't want to add the zeros, you'll get a much smaller download. And you will also get a metadata file, so you can add in any samples that didn't observe the species or didn't observe any fish. And you'll have a much simpler count file. On the download data and quality control score plot, you will see the quality control score plot. The number of segments will depend on the type of data that you have uploaded. If you've uploaded data from EventMeasure, your plot will have 5 segments. If you've uploaded generic data, your plot will only have 4 segments. The first segment in the plot is the green one for sample metadata, and this indicates if your sample metadata meets the criteria outlined in the requirements for the sample metadata. You can also think of it as the proportion of data that will be successfully uploaded into Global Archive. So if you get a score of 100%, all of your data is formatted correctly and all of your data will be okay to upload in Global Archive. If you get a score of 50%, only half of your rows are formatted correctly. You should aim for a score of 100% in the sample metadata. The next segment is the count score, and this indicates if all samples in your count data have a match in your sample metadata. If you're uploading EventMeasure data, your count data comes from your points, and if you're uploading generic data, your count data comes from the count file. You should be aiming for a score of 100% in your count score. Anything lower than 100% means that you have mismatches in your sample names. The next segment is the count versus length, and this indicates if your maxends are equal to the number of length measurements plus the number of 3D point measurements. If you have measured your campaign at maxend and you've added 3D points so that the total number of lengths and 3D points equals the maxend, then this score should be 100%, and you should be aiming for 100%. If you have not used 3D points, ignore this score. The percent length score indicates the proportion of measurements, so 3D points and length measurements, that have a value in length. If you have not used 3D points to ensure that every individual at the time of maxend has been accounted for, then ignore this check. If you have, you should be aiming for a score around 70%. And if you've got a score below 70%, it could indicate maybe you have low visibility, habitats obstructing the field of view, or you have other issues preventing length measurements, and you should look into this further. The length score indicates if all the samples in your length data have a match in your sample metadata. You should aim for a score of 100%, and if you get less than 100%, it means you have mismatches in your sample names. So either your opcode or your period, whichever one you have used in your annotation data, doesn't have a match in the sample metadata. And if you were to upload this data to Global Archive, it would get deleted because it doesn't have a match in the sample metadata.